Hi, everyone. My name's Gabe Haller. I'm the project lead for Project One, part of the BO1 that discussing this evening. I'm a PhD in the Department of Neurosurgery here at WashU. I study human genetics for the most part. And so I'm going to tell you a little bit of the nitty-gritty about what we've been doing to study the genetic underpinnings of Chiari malformation, and what we hope to continue doing by growing the cohort of patients and family members that we're collecting DNA from and analyzing go forward. In a relatively short amount of time, I'll tell you a little bit about what we're doing from a genetic standpoint, doing association analysis. And I know this is a complicated concept, but I'll try and make it as straightforward as possible. And then I'm going to tell you a little bit about how we're trying to understand what we're finding by analyzing the genetics of patients and their family members in zebrafish models here at WashU to understand the effects of disruption of certain genes on the brain and CSF and other neurological outcomes. So first of all, I'd like to thank anybody who's watching or here for participating in our genetic study here in the clinics at WashU and at various sites around the country. We've been asking people to donate their DNA for the study, and many people have been so kind to oblige. We make it as simple as possible, hopefully, to collect DNA by just using scope and getting saliva samples from patients rather than blood. And we will send out kits to your homes or have people come and talk to you in clinics when you happen to be at the hospital. And we also make it pretty easy. So if you want to click, take a picture of this QR code or anything to participate, you can, and it'll bring you to the Chiari website that Tanda mentioned, and it will link you to our study and the ability to have us contact you to participate. So over the years, we've been collecting DNA from a lot of people from various sites around the country. These are actually places where patients have sent DNA, but we don't actually have a site. So many places around the country have just contacted us. Patients on their own have come to our website, and we have sent kits for various places. We've been recruiting people for the genetics study since 2009 as part of the Park Reeds Consortium, but even before that, actually part of some of the scoliosis studies that we previously did because of the aforementioned connection between scoliosis and Chiari. But to date, we've collected DNA samples from over 1,300 patients and family members. So I'd like to actually very specifically point out that we have a lot of interesting studies that can happen when we have family members of patients with Chiari take part of this in the study. And so even though we only have, say, 900 actual Chiari patients, we have 1,300 DNAs from the patients and the family members that give us a lot of power in our studies. So not to belabor the actual science behind this, but and I thought people would be interested to know what we do. So when we get your DNA, we actually extract a portion of your DNA using these DNA baits that specifically target the portions of your genome that encode proteins. That makes it so that we can sequence many more people for the same amount of money. And we largely focus on these genetic variants that occur inside the proteins themselves. These are far more likely to be deleterious and thus also give us more statistical power to do genetic studies. We then take those capture portions of your genomes and we sequence them. Here at WashU, we have an amazing McDonald Genome Institute with many very powerful DNA sequencers that also make it relatively cheap to do because we have infrastructure to do that. We then call genetic variants in your genomes, which are all the places that differentiate you from everyone else in the human population. And then we try and use statistical methods to determine which of those is enriched in patients with Chiari versus patients without Chiari, or individuals without Chiari. So here's just an example of one of the things that we found. So these are actual genetic variants. In this specific gene, CHD3, CHD3 was previously associated with a number of conditions, but specifically those that have what's called macrocephaly associated with them. So this, again, ties back into what Dr. Lindbergh was just talking about, these different sort of subclassifications of Chiari. And so we found a number of individuals with deleterious genetic variants in this gene, which sort of implicate the idea of brain overgrowth, again, in Chiari malformation etiology. Another, again, important reason why we're interested in collecting DNA from parents of largely children with Chiari malformation is the ability to do statistical tests that look for genetic variants that occur in a child but not their parents. And of course, if both parents are still alive, at any age we can find these variants in a child or a person for and so what we're looking for, again, is these variants that are specifically in a child and not their parents. And those give us great statistical power to identify genes that are underlying a given condition. So we actually found a number of genes that harbored these kinds of so-called de novo variants. And one of them happened to be a very similar gene to that which we found previously, CHD3. And these are just a number of patients that were found to have these CHD gene variants. And again, they're occurring in the proband, or the patient with the Chiari malformation, but not their unaffected parents. We also found a number of other similar de novo variants in genes that were either known to cause diseases or known to be brain express, and they were enriched in these sorts of genes, these variants that were occurring in the kid and not the parents. So in this case, the gray line is significantly anything above this line is significantly enriched for that type of variant. Overall, we found a lot of different CHD genes, again, emphasizing this role of macrocephaly in Chiari malformation, potentially. Not to dwell too much on the statistics and numbers, but that led us to actually investigate the prevalence of enlarged head circumferences among patients with Chiari malformation. So here are two graphs of head circumference over time in the general population are the line, blue for males and pink females, and the dots are patients with Chiari malformation. And what we see is that for both sexes, the average distribution of dots for head circumference across all ages was increased in Chiari malformation cases. And some of them above the top line would be above the 95th percentile for age, and thus would be clinically diagnosed academically condition. But that being said, a lot of them are still within the normal range, but are above average, leading us to believe that at least some cases of Chiari malformation may be due to more subtle brain overgrowth in proportion to age. We also, I mentioned that we have been modeling these genes in zebrafish, and so we actually have zebrafish that are missing this gene, CHTA. And because of their transparent nature, zebrafish can be imaged quite readily using fluorescent microscopy. And so what we've done is image their brains. This is a zebrafish brain. You can see their large eyes and relatively different shaped brain than humans, but still we saw in every region that we looked at enlarged brain volume in the fish with CHDA knocked out, which is the minus plus in the middle. And so they're just overlaid so that you can see the normal zebrafish, we call wild-type zebrafish, in the magenta overlaid with the blue CHDA loss fish in the middle. So I hope that was sort of a taste of the kinds of research that we're doing. We're trying to collect DNA from as many patients as possible, identify genes that are contributing at least to certain individuals' Chiari malformations, and then understand how those genes are contributing to that by studying them in zebrafish.
It was clear. I'll be here for questions. Great. Thanks.